Welcome to AP Biology. Today I want to talk about translation with you. So we've already done some videos on transcription, which is DNA to RNA, and then RNA processing that happens in eukaryotes to make the messenger RNA. So today we're going to talk about how the RNA goes to a ribosome and gets translated into a polypeptide, which can then fold and become a protein. So first of all, the genetic code is a triplet code, which means that there are 3 letters of RNA that are read to make every amino acid. So these 3 letters in messenger RNA are called a codon. So the triplet code is non-overlapping 3 nucleotide words. A codon is part of mRNA that specifies a particular amino acid. So UGG will always code for tryptophan, and UUU will code for phenylalanine, etc. The codons in mRNA specify the order of amino acids in the polypeptide. So the DNA codes for the RNA. We read the template strand to make the complementary mRNA. And these 3 letters will code for an amino acid. 4 RNA nucleotides in groups of 3 produce 64 possible codons. So anyway, there's too many codons. And so we have this thing called a redundant code because the codon UUU codes for phenylalanine, and UUC also codes for phenylalanine. UUA and UUG code for the same amino acid. All of these code for the same amino acid. So there are more codons than there are amino acids. There's also a start. So AUG codes for the beginning of each polypeptide. The methionine might be cut out, or it might just be the beginning of the polypeptide, but they all start that way. And then there are 3 stop codons, which are written in here, so you don't have to memorize them. They don't signal an amino acid, they signal a stop for translation. So the genetic code is redundant. That means there's more than one codon for many amino acids. So these 4 codons actually all code for the same amino acid, leucine in this case. But it's not ambiguous, meaning CUU will always code for leucine. The codons have to be read in the right reading frame. So it's 3 at a time. These 3, then these 3, then these 3. It's nearly universal, which means this codon sheet works for just about every organism. Almost all bacteria and all animals and plants and fungi, et cetera, et cetera. So that means that we have a common ancestor who also used this code. So cool, right? So this is even cooler, I think. If you take a gene out of one organism and put it into another, it can still get expressed. So if you take a gene from a bug, a firefly, it's an animal, and you put it into a plant, if you put the glowing gene in and it gets expressed, you can make the plant glow, which means that plants use the same code as animals, and that they can recognize each other's code. So they did this first with one of the first ones was with fruit flies and mice. They had mice that couldn't see, and they took fruit flies that could see and took out one particular gene that had been missing in the mouse, and it was a little different in the fruit fly, but they inserted it from the fruit fly into the mouse embryo, and those embryos grew up with eyes. So we can read the DNA from other organisms if it's inserted into us. Here's a pig. It's pretty cool too. Here's a pig with with a jellyfish gene making it glow. So also cool. They're both animals, but very different animals, right? Vertebrate versus invertebrate. All right, so while gene expression differs among the domains of life, the concept of the gene is universal. And then another really weird thing, bacteria and archaea are both prokaryotes, right? They don't have a nucleus, they don't have all those internal membranes. But crazy stuff is that we, the eukaryotes, that's us, right? We share more DNA with archaea than archaea does with bacteria. So like how to make like a tRNA, that kind of thing. Archaea are actually more closely related to eukaryotes. We split a little bit later than the split between bacteria and the archaea and eukaryotes. So archaea share more features of gene expression with eukaryotes than bacteria, which is weird because they look more like bacteria. Okay, so vocab words and concepts that you're going to need to know. Transfer RNA. Transfer RNA or tRNA is the type of RNA that brings an amino acid to the ribosome. So it's folded over on itself, so it's transcribed as a single strand, but then it folds over into this strand. And so it's got an anticodon, and it also has a spot that picks up its particular amino acid. So each tRNA or transfer RNA carries one particular amino acid, and it has an anticodon on the other end. So this is a codon, it's part of messenger RNA, and this is an anticodon, it's part of transfer RNA. So you have to memorize codon goes with messenger RNA. The messenger RNA codes for whatever the amino acid order is going to be for the protein. And the tRNA just brings over the correct amino acid. So for example, this anticodon, CCG, I think it is, will always code for glycine. When you're reading the codon sheet, you're looking at the messenger RNA. So you're not looking at this, you're looking at this. So it's GGC that would code for glycine. But the way that the glycine gets to the ribosome is that the right transfer RNA with the right anticodon that's going to match up here will carry it. So the anticodon is in tRNA, it base pairs with the complementary codon. So if this codon is UUU, the anticodon would be AAA, and it would carry phenylalanine. So before you do any of this stuff, you've got to first match your tRNA with the right amino acid. So this is an enzyme that will connect the amino acid with the tRNA, and it takes some ATP, some energy to do this. And so here, first you phosphorylate the amino acid. That's what's happening here. So it takes energy to do that. And then the right tRNA is going to come in, and they get connected together. And now you have your tRNA ready to go to the ribosome. So that's sort of primed it. The next thing that happens is you have to have a correct match between the tRNA and the mRNA, so the anticodon and the codon. So the E, so there's 3 sites in the ribosome. This is the E where it exits, the tRNA that's done exits, and you've got your P site where the polypeptide is growing, and then you've got your A site where the next amino acid is going to come in. So this tRNA has its amino acid. They're going to be matched up here, so the codons to the anticodons, same with here, and then you're going to make a peptide bond between these 2 amino acids. So the second thing that happens is you have to have the right match between the tRNA anticodon and the RNA codon. Interesting thing here, this thing called wobble means that the first 2 letters always have to match, but the final one doesn't always match. So if you have CUC in your codon, the anticodon should read GAG, and leucine will be the right amino acid brought in. If you have wobble allowed, which happens for certain tRNAs, the CUU, it's only these 2 that actually need to match. This last one almost matches. It's called wobble pairing. You should have, let's see. So you should have, like, this U should code with an A. But with this tRNA, this is actually like a close enough. And so CUU actually ends up coding for the same amino acid as CUC. So flexible pairing in that third base only of a codon is called wobble, and it allows some tRNAs to bind with more than one codon. So CUC and CUU will actually get this same tRNA, which brings over leucine. Okay, ribosomes are made out of ribosomal RNA and proteins, and they actually help the tRNAs to fit into where they need to go in order to make the polypeptide that's supposed to be made. So the ribosome has 3 binding sites, the P site, the A site, and the E site. The E site is where it's exiting, the P site is where you have the polypeptide being formed, and the A site is where you have the new amino acid coming in. So there are 3 stages to translation. There's the start, there's the let's go with this, and then there's the final, the end. So initiation means start, elongation means you're making this whole polypeptide, and then termination means to stop. You also have these same 3 stages with transcription that we already talked about, but different things happen because transcription and translation are different, but they still both have a start, a middle, and an end. So for the initiation, we start with the ribosome not even assembled. So the mRNA comes on here to the smaller part of the ribosome, and you always start with methionine attached to its particular tRNA. This start codon is going to be AUG, and it the anticodon will be UAC, and then your other subunit, the larger subunit, is going to attach. And so then you've started, right? So the proteins called initiation factors bring the large subunit right here that complexes with the smaller subunit and the RNA with its first tRNA. Then elongation happens. So another tRNA comes over. It'll go to the A site where this amino acid will come. This is showing you that it's already gone through like the initiation here with the methionine plus 3 more. So this is sort of in the middle of elongation. And then you have a peptide bond formed right here. And that peptide bond is facilitated by RNA that's in the ribosome. So not every enzyme is a protein. The ribosome is actually working as an enzyme as well to make this bond right here. And it's actually the RNA that does that more than the protein. So most enzymes are proteins, but some enzymes are RNA. And here, this ribosome is an example of that. All right, and then the whole ribosome is going to move over because now this tRNA is done. It's going to leave, and the whole polypeptide chain is sort of going to be attached to this next tRNA. And so it's sort of this tRNA, right? Let's change colors. This tRNA right here is now going to be in the P site, and you're ready to have a new tRNA with an amino acid hop in here, and you just keep doing that until you're done. All right, so it starts on the 5' and goes toward the 3' direction. And then finally, termination or the end happens when you have the stop codon. There's 3 stop codons, UAG, UAA, and UGA, and you don't have a tRNA that has an anticodon that matches or that's complementary to these. Instead, you have this thing called a release factor, and when that happens, the whole thing stops. So the tRNA leaves, the polypeptide is cleaved, and now you have this free polypeptide which can finish folding and become a protein. In prokaryotes, translation of mRNA can happen at the same time. Transcription of mRNA can happen at the same time as translation happens. So translation can happen before transcription has even finished because there's no nuclear membrane to separate them out. Direction, the codons along the mRNA molecule are read by ribosomes in sort of the opposite direction that the mRNA is made. So it starts over here on the 5' to 3' direction. So that's the one that was first available. As you're continuing, I don't know if I'm saying this right, but you're continuing to add to the 3' side, but you're starting your translation on the 5' side. Polyribosomes just means that you can have a whole bunch of ribosomes on the same messenger RNA, all making their own, the same protein, but making many of them. If you need a lot of a certain protein, then this will happen. So you can make many copies of the polypeptide very quickly. Bacterial prokaryotic ribosomes are smaller than eukaryotic ribosomes, so you don't have to memorize any of this, but you can see that the subunits are a little bit smaller.